Hello and welcome dear viewers to the another informative video, and the topic is impurity types in pharmaceuticals and guidelines. So the video will cover mainly the impurity types, whatever the types of impurities are there in the pharmaceutical formulations and APIs, and the guidelines which are related to these impurities and their specifications and control. So first of all, little bit information we will see about the impurities. So whenever you have API or have a formulation in your hand and you think it's about its composition. So API is the active pharmaceutical ingredient, or you may call it as drug substance. So the drug substance is the main molecule which have the action or therapeutic activity, and that's why called as active pharmaceutical ingredient or the drug substance. And the formulation is made up of the active pharmaceutical ingredient or drug substance and the inactive materials or excipients. So then what is impurity? Impurity is any component that is not defined as active pharmaceutical ingredient or drug substance and the inactive material or excipient. Now, if you consider the drug substance, then what is an impurity? So, if you claim the drug substance, then whatever is present into that drug substance except the molecule of drug substance, that can be referred as impurity. Except the water content or LOD. And when there is a formulation, that time except the water content, you may say, if it is a solid oral formulation or liquid oral formulation, whichever is not claimed as a component of the formulation, that is referred as the impurity. And impurity is not an excipient and it is also not the active ingredient. Now, these impurities can come from manufacturing processes, degradation of the active material during storage conditions, from excipients, and or due to the contamination of the formulation or the active pharmaceutical ingredients. So the sources of impurities are manufacturing process, then degradation, storage condition, excipients, and the contamination. Now you might be thinking that, can we make the API or formulation without impurities? So the answer is no. The APIs or formulations cannot be made without impurities or 100% pure. Why? Because these impurities may come from any source like manufacturing process, there may be degradation on storage, there may be the excipients, impurities might be there, some impurities may come from the excipients or contamination. But the contamination can be handled through the total quality management system and the GMP practices. So contamination chances are very less. But the manufacturing process, degradation on storage condition, these cannot be avoided in totality. So we cannot make the API or formulation without impurities. Impurities will be there, but we have to control and limit the impurities above certain levels as given by the guidelines. The impurities affect the quality, safety, and have toxic effects also. Those are required and to be controlled. So the impurities are the main quality attribute of the formulation and the drug substance. These are the impurity is a critical quality attribute of the formulation. Then what are the types? The impurities are mainly divided into organic impurities, inorganic impurities and the residual solvents. So organic impurities and related substances. Some people call it as related substances. So organic impurities and the related substances are same or different. Now see, the related substances are structurally related to the drug substance. That means these are the API like. These substances may be identified or unidentified. That means may be known or unknown degradation products or impurities arising from the manufacturing process or during storage of the material. These are the organic impurities. Related substances are the organic impurities. The terminologies might be different or somewhat different at some extent, but the related substances are the organic impurities. So these organic impurities can be divided into different subtypes like process related, then degradants, mutagenic impurities, nitroso impurities or nitrosamines, including the NDSRIs, isomeric impurities or chiral impurities. So these organic impurities may be of different types. The process related impurities or process impurities come from the raw materials as intermediates, reagents, ligands and catalyst synthesis impurities and the from the purification step or from the synthesis step, and these are not the degradants. Then second is the degradants. These come from the degradation of API, degradation of API in the formulation, interaction between the API and the excipients. These also may come from during the manufacturing process or from the storage. So these are the degradants. Now some process related impurities are also degradants. So these are referred as the degradants. If it is a process related impurity, also the degradant impurity, then it should be categorized as degradant impurity and it cannot be treated as the process impurity. Then coming to the mutagenic impurities. These mutagenic impurities are those impurities which can cause the mutation, or these are the DNA reactive impurities and can change the DNA of the cell. Then nitroso impurities and the nitrosamines. These are also the organic impurities and the organic impurity molecule containing the nitroso functional group. These are referred as the nitrosamines. These are class 1 impurities, known mutagenic carcinogens as per ICH M7. So nitrosamines are the mutagenic impurities which are referred as class 1 impurities. Now these nitroso impurities or nitrosamine impurities are also can be categorized as small nitrosamines and the NDSRIs, nitrosamine drug substance related impurities, which we are going to see in the next slides. Then coming to the isomeric impurities or chiral impurities. See, some of the APIs are synthesized as racemic mixture or as the isomeric forms or isomer of the main compound. So isomer of similar compound have likely to own the dissimilar chemical or physical properties, and these similar or dissimilar physical and chemical properties can have some significant association, and this association or impact might be on the therapeutic side or from the degradation side. Then the mutagenic impurities we have seen, and some impurities have genotoxicity, some are mutagenic, so these have mutagenicity. Then what is the difference between the genotoxicity and the mutagenicity? Genotoxicity is the ability of the drug substance to cause toxicity on DNA or genetic material of the cell, while mutagenicity is the ability of the agent to cause mutations. And now you can relate this to the mutagenic and nitrosamines. Nitrosamine impurities may increase the risk of cancer if people are exposed to them above acceptable levels and over a long period of time. Nitrosamine can be highly genotoxic. So nitrosamines are the mutagenic impurities which are also the genotoxic. Then mutagenic impurities are also divided into some subclasses based on their mutagenicity and carcinogenicity. Carcinogenicity is the ability or property of the impurity to cause mutation and cancer. So class 1 impurities or class 1 mutagenic impurities are the impurities which have known which are known mutagenic carcinogen, and these are controlled at or below the compound specific acceptable limit. The nitrosamine comes into the class 1 type of mutagenic impurities. Then class 2, known mutagens with unknown carcinogenic potential. These are mutagens but their carcinogenic potential is unknown. Bacterial mutagenicity is positive and no rodent toxicity data is there. No rodent carcinogenicity data is there. And these are controlled at or below the acceptable limits. That is appropriate TTC. TTC is the threshold for toxicological concern, and generally TTC is the 1.5 mg per day. Then coming to the class 3, these have alerting structure unrelated to the structure of the drug substance, no mutagenicity data, and are required to be controlled at or below acceptable limit, appropriate TTC, or conduct the bacterial mutagenicity assay. And now if this class 3 impurity is non-mutagenic, then class 5, then this impurity will go to class 5, and if it is mutagenic, it will go to class 2. If it is going to class 2, then it is required to be controlled at or below acceptable TTC. And if it is going to non-mutagenic category, class 5, then it will be treated as non-mutagenic impurity. Then coming to the class 4 and 5 type of mutagenic impurities. So class 4 is alerting structure. The same alert in the drug substance or compounds related to the drug substance. Example, process intermediates which have been tested and are non-mutagenic. So treat it as non-mutagenic impurity. Then class 5, no structural alerts, no alerting structure with sufficient data to demonstrate lack of mutagenicity or carcinogenicity. So these are treated as non-mutagenic type of impurities. Now coming to the TTC. What is TTC? TTC is nothing but the threshold for toxicological concern. And the threshold of toxicological concern, that is TTC, is a science-based approach for prioritizing the chemicals with low level exposure that require more data over those that can be presumed to present no appreciable human health risk. This is the simple definition or elaboration of the TTC. Then coming to the nitrosamine type of impurities. These impurities are the impurities with nitroso group or NNO functional group. The classification based on chemistry, we can divide these nitrosamine impurities as small nitrosamines, mostly originating from the synthesis route, and the complex nitrosamines. These are the bigger type of or larger molecule impurities. These are known as drug substance related impurities, NDSRIs, and these are mostly originating from the synthesis route. So these are the impurities with N double bond O, and the small nitrosamines are NDMA, NDEA, NMBA, NMPA, NIPA, NDIPA with their acceptable limit. These have very low acceptable limit, 9 nanogram per day, with the lowest and AI limit of 26.5 nanogram per day, and highest for these type of impurities is 96 nanogram per day. Nitrosamines are the most potent type of impurities. Now coming to the nitrosamine drug substance related impurities. So the NDSRIs, which are subcategory of nitrosamine impurities, that share the structural similarity to the active pharmaceutical ingredient in the drug product, typically lack compound specific mutagenicity and carcinogenicity data to inform safety assessment. So on the basis of read across approach or SAR, structure activity relationship, these are categorized, and these have given the acceptable limits. So the examples I have given here, like N-nitroso-bupropion, N-nitroso-cinacalcate, N-nitroso-sertraline, N-nitroso-desmethyl almotriptan, and these have AI limit of respective to the molecule, which range from 1500 nanogram to 400 nanogram to 100 nanogram and 26.5 nanogram per day. And if the limit of acceptable limit or acceptable intake limit of 26.5 nanogram, then you can categorize this as small nitrosamine type of impurity, which is also having the same acceptable intake limit of 26.5 nanogram per day. That means this impurity is most potent type of nitrosamine impurities. Then the categorize, these have category 1 with AI value of 26.5 nanogram, category 2 with 100 nanogram, category 3 400 nanogram, category 4 1500 nanogram, and category 5. Category 5 is also having the 1500 nanogram per day as acceptable intake limit. Then coming to the isomeric or chiral impurities. See, isomers are the identical molecular formulas but different arrangement and the activities. Isomeric impurities are generated because of the conversion within the critical process parameters during the drug synthesis. So CPPs are involved, or CPPs are responsible factors for this isomeric or chiral impurity formation. So the chiral impurities or isomerism we will not see in that much detail, but here we will see the types of isomerism, like structural isomerism may be there or stereoisomerism may be there as subcategory. Optical isomerism is the most prevalent for these drug molecules, like levorotatory, dextrorotatory, you might have heard somewhere or studied. So the example is omeprazole molecule or isomeprazole molecule. Some molecules are present as racemic mixture, like atenolol is there, warfarin is there. Then some molecules have chiral structure, chiral carbon is there, like omeprazole and cyclophosphamide are the example of drug substance. So these chiral impurities in pharmaceutical samples may occur as side product of the synthesis process as a result of the inversion of chiral centers due to chemical degradation of the drug substance or both. So these chiral impurities may come from the process, or sometimes these may get formed as degradant impurities. So based on the process or degradant type of impurities, the specifications are required to be given. Then coming to the inorganic impurities and residual solvent. These are the two different type of impurities apart from these organic impurities. So in organic we have seen the different type of impurities. Now we will go to the inorganic and residual solvents. So in inorganic impurities, mainly these come from the process, because generally these are inorganic impurities or metals are not used into the manufacturing process, or there are very less chances to come from the equipments. So reagent, ligand, catalyst, heavy metals and residual metals, inorganic salt, filter aids or charcoals. Use of charcoal sometime is there in the process. So these elemental impurities come from there. Then residual solvents. Solvent present in the API or formulation from the manufacturing process. Solvents may come into API from the synthesis route, or from in the formulation from the raw materials, and if these residual solvents like acetone, IPA, ethanol, methanol, these are used into the manufacturing process, they may come from that manufacturing process as well. The residual solvents limit specifications are required to be given if solvents are used into the manufacturing process. If solvent is used into the manufacturing process, then that is required to be limited and the specific limits are required to be given. Then inorganic impurities subclasses we will see. These are coming from the process mainly. Class 1 impurities are the mercury, lead, cadmium, selenium and arsenic. These are mainly the human toxicants. Arsenic is there, cadmium is there, mercury is there and lead is there. So these are the human toxicants and these have limited or no use in the manufacturing of pharmaceuticals. Then class 2. These are the classes generally considered as root dependent human toxicants. Class 2 elements are divided into class 2A and 2B depending on their relative likelihood or abundance into the drug product. Then class 2A, these are cobalt, nickel and vanadium, and class 2B, these have reduced probability of occurrence, and the elemental impurities include Ag, Au, Ir, Os, Pd, Pt, Rh, Ru, selenium and Ti. Then class 3. Class 3 are relatively low toxicants and by oral route of administration their PD is generally less than or sorry their PD is higher than 500 microgram per day. And other elements are also there uh, which have not uh, the established not having the established toxicity due to their low inherent toxicity, and there are some differences into the regional regulations, so these are not given with the proper PDEs in the ICH guidelines. Then residual solvents. Residual solvents are used into the manufacturing of the drug substance and the drug products. These are used into the formulation as granulating aid or granulating solvent or coating material solvents, or sometimes these are used to dissolve some materials and then that dissolved form of material is taken into the process of manufacturing of the formulation. So class 1 are the solvent to be avoided because these are carcinogenic, toxic and have environmental hazards. Examples are benzene, carbon tetrachloride and many more. Then class 2 solvents, these are to be limited because these are toxic. These are like methanol, toluene, cyclohexane, chloroform, acetonitrile. Then class 3 solvents are there. These all are the impurities. These are considered as impurities into the drug substance and the formulations. And these are required to be removed and some steps are required to be taken to remove these solvents if used into the manufacturing process. So class 3 are the solvents with low toxic potential compared to the class 1 and 2. These are acetone, ethanol, isopropyl alcohol and ethyl acetate. Many more examples you can find in ICH guidelines. Then the guidelines for the impurities. Mainly the ICH guidelines are there which are dealing with each and every type of the impurities. So impurities in new drug substances is Q3A. Then impurities for drug products it is Q3B. Guideline for residual solvents you can see Q3C guideline, and for elemental impurities you can see impurity guideline Q3D. Then ICH M7 is the safety guideline which is into the miscellaneous type of guideline for assessment and control of DNA reactive mutagenic impurities into the pharmaceuticals to limit the potential carcinogenic list. Then for nitrosamine impurities you can find many more guidelines in by the published by the USFDA and the EMA. And also these guidelines are updated always, and these are published by the different health authorities like ANVISA is there, EMA is there, Swiss Medic is there, USFDA is there. So you can go through those guidelines and can have a detailed understanding from these guidelines. So this is about the impurity types and their guidelines. So many more videos are there into the Pharma Learning in-depth channel. You can go through these videos and have in-depth understanding about the impurities. The reason for making this video was to have a complete understanding about the types of impurities and the guidelines. So the next upcoming videos will be for each and every type of impurities, like elemental impurity calculation, uh, specification for residual solvents, specification for organic impurities, specification of nitrosamine impurities, and many more. So keep watching these videos to have good understanding, and I hope if you watch these videos uh, repetitively, you will get the correct information and have a good understanding about the topics which are very much required for the pharma professionals. So thanks for watching the videos, and till then, take care and bye bye. Meet you soon with such kind of informative videos. Thank you.